Yeah, everybody for the posters has 1 minute. And then the next speaker can immediately take over.
Awesome, great. All right, hello everyone. So I'll try to be brief as possible. So in this study, we want to evaluate the reported levamisole resistance marker ACR8B in Haemonchus contortus. So briefly speaking, we used adult worms from 3 phenotypically resistant, 3 phenotypically susceptible, and 3 Swedish susceptible to levamisole isolates, as well as larvae from these Swedish farms where levamisole was considered to be effective. Next slide, please. All right, so essentially what we did, we created a ddPCR assay, and we found that the 63 base pair deletion that happens in the ACR8 gene is actually way more common overall than not. And even though statistically speaking, there's a genotype difference, and these are significant between the susceptible and resistant isolates on rows 1 and 2 on the left, the ones that are marked S and R. But the deletion containing allele is overly abundant, even in susceptible isolates, essentially. Now, in the larvae on the right hand side of the screen, different frequencies of the deletion containing allele were found in the pretreatment samples. However, the treatment was effective nevertheless, as measured by the fecal egg counts, as well as ddPCR. So overall, this casts some doubts about the utility of this deletion as a resistance to levamisole marker in the field context. Thank you very much.
Okay. Thank you for the opportunity to present my project. My name is Paula Ehnert from the Freie Universität Berlin. In attribution to recent studies on anthelmintic resistance in Europe, we look closer on the current resistance situation in northeastern Germany and Brandenburg. And we can provide initial findings of a fecal egg count reduction test that has been conducted on 11 sheep farms using the drugs fenbendazole, ivermectin, and moxidectin. Results were achieved using mini FLOTAC with a 5 eggs per gram sensitivity and analyzed using the R package egg counts. Results can be seen on the second slide of my presentation. Reduced efficacy was found for fenbendazole and ivermectin on each 6 of 8 farms tested, plus 1 farm with suspected ivermectin resistance, while moxidectin resistance was detected on 2 of 11 farms. The perspective of our study is to do the same fecal egg count reduction test on cattle using fenbendazole and ivermectin in spring 2021. And the collected material will be used to conduct a larval migration inhibition assay using levamisole and to determine nematode communities before and after treatment using the Nemablum next generation sequencing approach. Thank you very much for your attention.
Hi to everyone. And this study was performed in collaboration with the Gent University, and the aim was to perform a first validation of the Kubic FLOTAC microscope on fecal egg count of gastrointestinal nematodes in cattle. You know that the KFM is a new digital microscope that combines high sensitivity, accuracy, and the precision on FLOTAC and FLOTAC techniques with a label system to capture and analyze the pictures. Please, the second slide. In this study, we collected 30 positive fecal samples from cattle experimentally infected by genes in Gent, and in lab, we have prepared 6 subsamples for each fecal samples, and we have used 2 different reading systems, a traditional optical microscope and the KFM. And the results showed a substantial agreement with a low discrepancy, as you can see in these 2 graphs. And so the KFM is a promising tool for an accurate and efficient fecal count and to perform remote diagnosis as expected by teleparasitology. Thank you.
Good afternoon, everyone. I am Marieke van Akke from Gent University, and I've been doing some research into risk factors for infections with Dictyocaulus viviparus in dairy cattle and also in associated production losses on Flemish dairy farms. So what we found is that farms have a high risk for an outbreak when the bulk tank milk ODR is above the cutoff of 0.41, especially when this is the case in 2 consecutive samples and in August or October. Also, the risk for 2 consecutive positive bulk tank milk samples was higher when a farmer frequently purchased new animals and when a higher percentage of the grazing season was covered by anthelmintic treatments. The risk was lower when at least 50% of the pastures was mowed, comparing to not mowing at all. Second slide, please. Linking each test day of the milk production to the closest bulk tank milk sample, we found a loss in the mean milk production of 0.26 kilograms per cow per day, and a loss in the mean percentage of fat of 0.02% over the interquartile range. There was also a loss in mean milk production, mean percentage of fat and protein when farmers had 1 or at least 2 consecutive bulk lamella samples above the cutoff of 0.41 ODR. Thank you.